Yep.
Okay.
So we're recording now. I'm going to go ahead and ask everyone to go ahead. Well, actually, I'm going to hit the mute all here. And just remember, Michaela, you'll need to unmute yourself, okay? And you can take it away with your introduction slide and a brief introduction of who you are, just recording, okay?
All right, sounds good.
All right, I'm muting everyone now.
Oh, can you re-enable the screen sharing? For whatever reason, it took it off.
Yep, one moment, sorry.
It's okay.
There we go. You should be now.
Yep, that should do it. All right, can you see that okay? All right, perfect. All right, well, thank you for that introduction and thank you for joining us this morning for my thesis defense presentation. I'm going to be presenting my work over the last several years, which is entitled Assessment of Indiana Bat Reproductive Condition, Recapture Trends, and Temperatures of Artificial Roosts in Kentucky. So I'm going to give a brief overview of how we're going to run through this talk. I have 3 sections in my thesis, and the first chapter is entitled White Nose Syndrome Impacts on Reproductive Recovery of Indiana Bats in Kentucky. And this is really the principal research objective in my proposal and the main reason that this research was funded. Let's see. And then we're going to go into chapter 2 and 3. We were able to extend this project into a couple different avenues here. And we were able to look at recapture trends in these Indiana bat colonies, as well as looking at temperature conditions of artificial roosts that these bats were using. So before we jump into chapter 1, I'm going to give a brief overview of my study species, which is the Indiana bat or Myotis sodalis. Myotis is a mouse, or means mouse-eared, so this is a mouse-eared bat. And sodalis in Latin means companion. And you can see that these bats are clustered together in this group here. Indiana bats have a characteristic trait that in caves they will be clustered together, sometimes by the thousands. So they are known as kind of a social bat. And this is a federally endangered bat species. And the reason for its original listing partially was due to winter hibernacula disturbance, where people would head in or would destroy entrances of caves or alter the cave environment, which can change the temperature or air conditions, as well as summer roost habitat loss. These bats live in snags or dead or dying trees in the summertime, and the reduction of these from improper forest management techniques also led to the species decline. So over on our right-hand side, we can see the overall range of the Indiana bat. And you can see that Kentucky really is right in the middle of the Indiana bat's range. And we're also in this blue Midwest Recovery Unit, which is part of the overall recovery plan for the Indiana bat as a species. So with that, I'm going to get into chapter 1, which is entitled White Nose Syndrome Impacts on Reproductive Recovery of Indiana Bats in Kentucky. Again, this is the first chapter and the largest overall component of this thesis. So first we need to talk about white nose syndrome, which is Pseudogymnoasticus destructans, or a fungal disease that impacts 13 species of bats in North America. So if you look over at the bat on our left-hand side, you can see these patches of fungus on the bat's arms, and then one kind of on its ear. There is some on its nose as well. You can't really see because of the glare of that photo, but the white nose syndrome, or the fungus on the nose of the bats, is kind of where white nose syndrome gets its characteristic name, because the bats have white noses with this fungus on them. And so this fungus depletes the energy of bats because they're irritated by this fungus. They'll arouse from torpor more frequently during their winter hibernation period. And while the fungus itself is an issue, the greater issue is they are depleting all of these energy reserves because they're irritated and waking up more often. And this can lead to essentially bats starving because they just don't have this energy resource conserved to make it through the winter with the additional stress of white nose. There's an additional issue where they will have to immediately jump into a healing and immune response come the summer period or spring period when they exit the caves, which can be particularly taxing on female bats, which have to jump right into their reproductive cycle come spring. This is a map that shows the first county where white nose syndrome was discovered in 2006 in the United States. So this is in Schoharie County, New York. And as you can see by this map moving forward, the fungus from there spread throughout the eastern United States up into Canada. And then this is a screen capture from the 2011 year where white nose syndrome was discovered here in Kentucky in Trigg County. Jumping ahead to the most recent spread maps of white nose syndrome, you can see that it's spread across the United States and over to the West Coast. In our area, white nose syndrome really is so widespread that most caves in Kentucky have likely seen some impact from this fungus. And so we know that female bats have to be necessarily thrifty with energy or more conservative than their male counterparts because, again, they're jumping into that reproductive cycle come spring, so they have to have a little bit of reserves left over anyway. But research on the subject of white nose syndrome and reproduction has reported instances where females appear to be foregoing pregnancy or losing pups, and surveys have also reported instances of lower juvenile captures. Overall, while more research has reported reproductive potential in some white nose syndrome impacted Myota species, one of these being a lab study, there is still a lot of uncertainty to the degree around which reproduction is affected by white nose syndrome. So that brings us to the main reason that this research was funded and our main question for this research, which is if bats are entering into a disturbed hibernation period, they're coming out in the spring with depleted energy, are females able to carry a pregnancy to term, or are they going to forego pregnancy or lose that pup? And so that brings us to our first objective, which is to investigate current reproductive trends in white nose syndrome impacted Indiana bat colonies in Kentucky. And we followed that up with another objective, which was to compare the reproductive capacity of pre and post white nose syndrome adult female bats in the maternal season in Kentucky. So you've probably noticed by now that this study has a very much Kentucky focus to it. Our state had the 4th largest overall net loss of the Indiana bat since 2007 in comparison to other states. I think we've lost about 15,000 Indiana bats is the current estimate. And over here on our pie chart, this represents where the overall Indiana bat population, as far as the range-wide surveys went in 2019, where the majority of these bats were located in these overwintering hibernacula. So Kentucky contained 10.4% of the overall species overwintering in caves in the winter of 2019. Our focal sites for our study were also Kentucky Fish and Wildlife, or KDFWR, Indiana bat focal sites. And 2 of these were wildlife management areas, while 1 of these was a partner site. And it's important to note that artificial roosts were present at all of these sites prior to our study, which is a habitat management tool and something that we were able to use in this study as part of our research. So for my 3 study areas, we have Yellowbank WMA over to the left in Breckenridge County, Kentucky, Bernheim Forest in Bullitt County, Kentucky in red there, and then Veterans Wildlife Management Area in Scott County, Kentucky. And it's important to note that Veterans WMA has 3 distinct clusters of roosts that Indiana bats are using there. And so we'll talk about that a little more as we move forward. For our capture technique, in order to catch bats as they are exiting the roost at dusk, we conducted a pre-parturition survey June 1st through 15th, and this was to look for whether bats were pregnant or lactating, what their reproductive status was in these colonies during this time. And then we also conducted a post-parturition survey window in mid-July to check for juvenile recruitment in our colonies. We netted 1 roost per site visit, and that's what this looks like in this picture here. This is our artificial roost, so bats would fly out at dusk. And to capture them, we had 2 4-meter nets set around the roost, along with 1 6-meter net around the back. And we also we placed a stake in the ground about right here. So we could lift up this pole and place it back on the stake and kind of open up that triangle if we needed to cut off captures for whatever reason that evening. We did gain EKU, IACUC, Kentucky Fish and Wildlife, and US Fish and Wildlife Service approval for this project. We followed white nose syndrome decom protocols. And this research was conducted under Kentucky Fish and Wildlife Section 6 permits. We did have plenty of people, vaccinated volunteers that we were extremely grateful for that came out and helped us make sure we could get bats out of the nets in a timely fashion. And we also had permitted biologists on site for every netting evening. So to look at the morphometric data that we were collecting with each of these bats, we looked at the reproductive condition of females, so whether they're pregnant, lactating, post-lactating, or non-reproductive. We looked at the age of the bats, whether we were catching juveniles or adults. We took a measurement of their mass and then right forearm length, which is a standard measurement taken for bat size. And that's what you can see in the picture on the left-hand side. We also took a wing score for white nose syndrome damage. That's a 0 to 5 rating, with 0 being the best and 5 being the worst. And we did a UV light check to see if white nose syndrome lesions were on these bats. They would fluoresce if they were present, but we did not find any white nose syndrome lesions. We didn't have any bat turn up positive with that check, likely because we were netting during the summer period and white nose syndrome lesions tend to clear up in that summer heat. And all adults were banded before release. So to give you an idea of our sample effort across the 3 years, in 2017, we had 6 netting nights. This bumped up to 8 netting nights in 2018 with the conjunction of EKU joining the project and having more help. And then we also continued to do this for 8 netting nights in 2019. In 2017, we captured 209 Indiana bats, 1 big brown bat and 1 evening bat. And then in 2018, we caught 327 Indiana bats with 1 big brown bat, which followed very similarly in 2019, capturing 330 Indiana bats and 1 big brown bat. We had 206 bats that were banded in 2017 with 2 recaptures. We had 236 bats banded in 2018 with 60 recaptures, and then in 2019, 276 banded bats with 57 recaptures. So across the 3 years of this study, we had 870 total bat captures, with 866 of these being Indiana bats. And I am happy to report that all of these bats were released unharmed after our capture evenings. So moving into our analysis here, we were looking to assess white nose syndrome impact on female mass. And so female mass here was serving as an indicator of health in these colonies, the health of bats in these colonies. So to do this, we modeled adult female mass using both body condition and white nose syndrome factors. And our response variable here was mass of female bats. We also had a slew of predictor variables, and our reproductive class and RFA in pink here are what are our body condition factors that we were considering to be kind of our business as usual, or these would be affecting the mass of female bats in our populations, whether or not white nose syndrome was present. Down here, our wing score and year are both our white nose syndrome factors. So again, with that wing score, as it's going up, that there's more impact on the bat of white nose syndrome. So we suspected that bats would have lower weight with this impact, as well as they're entering these caves in the wintertime year after year. So we suspected that they may continue to decrease in mass over consecutive years. We also modeled Julian date of their capture and then site as random factors. Site, we considered that bats had been exposed to white nose syndrome at all 3 of our capture sites, so we weren't trying to dig into the individual differences there. We conducted AIC model selection with linear mixed models that we had created, and we did square root transform these variables to fit normality. And so moving on to our model ranking, you can see that our year model, our body condition model, and our wing score model all had a delta AICC of less than 2. And so these were all 3 competing models in our analysis. So we conducted model averaging to pull out our significant parameters. And as we had suspected, pregnant bats and bats with larger forearm length did have significantly higher or significantly greater mass, which is kind of what we expected with those business as usual factors. We also saw that wing score of 1 had a negative impact on bat mass, and then year of 2019 also had a negative impact, but only when compared to reference level year 2018. You'll notice that we did use 85% confidence intervals for the significance here, and this is due to this Arnold 2010 paper, which discusses the compatibility of 85% confidence intervals with AIC model selection, more so than the narrower 95 and 90% confidence intervals. So to pull out some of those informative parameters and start looking at them individually, we have adult female bat mass on our y-axis here, and then years 2017, 2018, and 2019 on our x-axis. So you can see the spread of adult female bat mass throughout our 3 years. And bat mass did seem to remain consistent. We're really not seeing that drop between the 2019 and 2018 years. Mass really did kind of seem to maintain consistency throughout the 3 years of our study. So here we're moving on to looking at individual wing scores. Again, we have our massive pregnant female Indiana bats on our y-axis, and then the date of capture on the x-axis. We've got our wing score, either a 0, 1, or 2, and then which year it was captured in. And so these are all of our pregnant bats throughout the 3 years. And you can see that these wing scores of 1 in gray here are very equally distributed throughout our captures. There's not a huge deficit in bats that are really low weights compared to ones that are really high weights that have a wing score of 1. We did have our wing score of 2 kind of hidden in here. It's behind these other points on the plot here. But we're not seeing a huge drop in those wing score of 1 weights. Lactating bat mass came out similarly. So our lactating bats here, again, with our wing scores of 1, we've got them really kind of both on the upper and lower end of mass. It does look like our bats are decreasing, lactating bats are decreasing across the season, but this is more than likely because here we're catching lactating bats that have just come off of pregnancy, and down here this is in July, so they've had some time to come off of that main pregnancy weight. Moving over to our non-reproductive bats, we did see a couple data points here where non-reproductive bats seem to have a lower mass at the wing score of 1. We didn't have a huge amount of overall non-reproductive bats captured in this study, which was really positive to see, but we may have been able to better evaluate that if we had had a higher sample size of non-reproductive bats. Moving over to our pre versus post white nose syndrome reproductive capacity analysis. So we're looking here to figure out if our bats post white nose syndrome that we're catching in our study are differing in their reproductive capacity from our pre white nose syndrome bats. So in order to do this, we took pre white nose syndrome records, which were retrieved from the Kentucky Fish and Wildlife Bat Database. And these ran from year 1979 to 2010. The year before white nose syndrome was discovered in Kentucky, and ran between June 4th and July 17th. It must be acknowledged that this is a very limited data set. We had to call out a lot of records which were incomplete or just incompatible for use in this analysis. But this was every single record that KDFWR had, so it was the best that we could do as far as gleaning this pre-white nose syndrome Kentucky data. For our response variable here, we used reproductive proportion, so whether the bats were reproductive or not. And then our predictor variables were white nose syndrome status, either pre or post white nose syndrome. And then county was modeled as a random factor because in this in the modeling that we were doing with this binomial distribution, we needed to have a way to break up the different, our different variables here. And so we conducted a generalized linear mixed model, again, with that binomial distribution, either reproductive or non, and we used an ANOVA to compare this model against the null. And what we found was that there was no difference between our pre and post white nose syndrome proportions. And we also looked at our post white nose syndrome study 2017 through 2018, and we didn't find a difference between any of our years post white nose syndrome. So to give you an idea of what that looks like, pre-white nose syndrome over here is at 99% of captures were reproductively viable. And then moving into when our study started in 2017, there was 89% of bats were reproductively viable in 2017. 90% of bats were reproductively viable in 2018, and then 96% in 2019. So these proportions did not differ significantly, and there is not much of a difference. There is a little bit of a drop there between the 99% and 89%, but again, no significant difference between those proportions. We also wanted to look at how pre and post white nose syndrome periods would differ between our pre-white nose and post-white nose bats. And so pre-white nose is up here, post-white nose is down here. We have pregnant bats, lactating, and post-lactating bats. And these are the time lengths where the first bat that was pregnant or lactating was caught, and then kind of the end time length where the last one was caught in each of those data sets. So what you're seeing is these patterns followed similarly. There wasn't a huge amount of difference in post-white nose syndrome bats having stages that were a lot earlier than our pre-white nose syndrome bats. We are kind of missing, it does seem like post-white nose syndrome bats we didn't catch as many that were pregnant towards the end of June, but that is because we didn't sample in the end of June in our study due to disturbance concerns at those artificial roosts. So I am really happy to report that reproduction is sustained in our study colonies. Our pie chart to the left shows all of our adult female bats through the 3 years, and 92% of these adult female captures showed signs of reproduction in our study, with only 8% that were non-reproductive throughout the period. Over here on our blue pie chart, we caught 55% of our July captures out of 307 total July captures were juveniles. And so we are seeing that juvenile recruitment in the population. We're also seeing recapture bats, so our adult females that we caught on one instance and then we caught another instance over here in our left pie chart. And so 89% of those several capture females were reproductively viable at both captures. Less than just under 10% were non-reproductive at first capture and then became reproductive at the second capture, with only 2% non-reproductive at both captures. And then over here on the right, we had 8 bats that we captured as juveniles for the first time. And so we captured them as juveniles, and then when we captured them the second time, they were reproductively viable. And so we're seeing that not only are females in our colonies able to produce offspring, but these offspring can then grow into adults that can produce viable offspring themselves. Our conclusions for this section are that wing score of 1 had a weakly negative impact on bat mass in our white nose syndrome model, which did follow our expectations for the study. Contrary to our expectations, our reproductive percentage of post-white nose syndrome captures did not differ significantly than our pre-white nose syndrome captures. And despite evidence of white nose syndrome impact, reproductive capability is sustained in our colonies, which are very promising results. So we're moving over to chapter 2 here. And this is entitled Recapture Trends in Maternity Colonies of Indiana Bats in Kentucky. So a brief introduction here. Recapture rates in most bat studies are generally quite low, and we have a unique study here because of our intensive capture effort that allowed us to have site visits across the 3-year period. So we wanted to kind of take a deeper dive into the recapture data. And our objectives for this section were to investigate bat movement within and from our capture sites, to determine recapture rates at our locations, and then also to explore recapture reproductive trends for use in our greater data set. Just a note here that these data were pulled from the exact same study that I just talked about, so the methods are the exact same as the ones we went over for chapter 1. So our overall recapture rate across this 3-year period was 14%. This is 119 bats out of 870 captures. In 2017, 1% of our captures for the year were recaptures. This jumped up to 18% in 2018, and then stayed pretty consistent in 2019 at 17%. When you look within our study sites, we're looking individually at Veterans WMA, so within your recaptures here, you can see that by far the majority of our recaptures are occurring in either the early June or mid-June period here. This is both for 2018 and 2019. And then once we get into July, those recaptures for the year start to drop off. And this is because most of what we're catching in July is juveniles. So they, of course, have not been caught or banded yet. At Bernheim Forest, this trend followed similarly. We had the majority, again, of our recaptures in June of both years, and then in July to a lesser extent. We do kind of see more recaptures in July of 2019, and I think this is because the colony at Bernheim is a lot smaller than the other two colonies, so we do have some of those male bats that, even though they've kind of left the maternity roost, they seem to be coming back year after year with the colony. At Yellowbank WMA, we also saw more recaptures in June than July. And then in June of 2019, we actually had 100% of our recaptures for that year occurred in June and none at all in July. And so while we're going to these sites and trying to net the same roost every time, these bats are kind of switching. And this kind of documents movement here, because although we may have netted the same roost, the bats have likely moved on somewhere else or have kind of switched scenarios for a little while. And so we don't always catch the same bats at every single time. At Veterans WMA, we were able to take a closer look at some of the roost switching and cluster switching between the three clusters of roosts there. We have our site of initial banding, which is either our north, central, or south site, and then the site of where the bat was recaptured, again, north, central, and south. So we saw our greatest areas of overlap between our north and central site, and then again at our south and central site, we believe that the central site kind of serves as an intermediate area between that north and south site. So bats are kind of moving in between all three, but possibly using the central a little bit more. But high is relative as far as all of these percentages go, because they're all quite low. And the sampling effort was higher in the north section than it was at the other two. So we may have actually seen a bit more overlap had we been able to sample the central a little bit more. We did have 2 occurrences of bats that were captured at all 3 veterans roost sites. So we're seeing bat movement between all 3 of our clusters there. So we were also able to see some interesting recoveries in winter hibernacula within the state. And so this is our blue, we're gonna pretend this is our bat here. And we had connections between Yellowbank WMA and then B&O Cave in Breckenridge County, in the same county there. And that bat moved back to Yellowbank WMA. That shows that we had a female bat that was captured prior to and post hibernation in B&O Cave, which is a white nose syndrome positive cave. And that female was reproductive on both occurrences, which was interesting to note. For Bernheim Forest, we had a recovery at Jesse James Cave, primarily a gray bat hibernacula, kind of in that same Mammoth Cave area. And that was down in Edmonson County, Kentucky. We also had 2 bats from Veterans WMA that went over and hibernated in both, or one each in Bat and Saltpeter caves. These are in Carter County, Kentucky, and these are both priority 1 Indiana bat hibernacula, so important to note those connections. So bats within state traveled between 27 to 125 kilometers. And so we know that bats are not looking at these state lines and realizing that they're crossing them, but this is more of just a way to better organize where our bats are moving for us to kind of keep a hold of. And so for our out-of-state captures or recaptures, we had some that were recovered by Indiana DNR in Indiana. So we had connections from Yellowbank WMA to Wyandotte Cave in Crawford County, Indiana. We also had connections from Yellowbank WMA to Jughole Cave. Again, that bat's moving back to Yellowbank WMA. That's another female that was reproductively viable pre and post capture in Jughole, or pre and post hibernation in Jughole Cave. And Jughole Cave is really important to note because it is a really large Indiana bat hibernacula. It was the 2nd largest in the last 2019 range counts, and it is presumed to be white nose syndrome positive. Bernheim Forest also had a connection with Jug Hole Cave where bats were recaptured there. And then Bernheim Forest also had bats that were recovered in Wallier Cave in Harrison County, Indiana. So bats traveling out of state traveled between 42 and 66 kilometers. And this is really important to note because looking at our top 10 largest Indiana bat hibernacula, these are of the 2019 range counts, we have four of our caves where we've had recoveries from these study colonies. These are starred here. We're in the top 10 largest Indiana bat hibernacula as of those 2019 counts, and 3 of them are in those top 5 hibernacula. So we have some really important connections with these large Indiana bat hibernacula. I mentioned that Jug Hole Cave was one of the larger ones. That's almost 80,000 bats counted in 2019. So our conclusions for this section, we had a banding rate of 82% and a recapture rate of 14%. We had bats that traveled 125 kilometers. That was the furthest recorded in-state migration. And we also had bats out of state with the furthest recorded migration of 66 kilometers. And you can see that our recapture rates have varied widely between sites here. We also had 80% of our female hibernacula recaptures that had a known reproductive condition that showed signs of reproduction at their initial banding. And so we have bats that are potentially coming out of caves affected by white nose syndrome, are going through those energetic expenditures of pregnancy, and then are still able to complete that migratory cycle. We also had 2 females that exhibited that reproductive capability both prior to and following their cave recovery in either white nose syndrome positive or white nose syndrome, or assumed to be white nose syndrome positive caves. So that brings us to our 3rd chapter, which is temperature conditions of artificial roosts used by Indiana bats in Kentucky. So we know that Indiana bats preferentially roost under the shedding bark of deteriorating snags, and that's an example of what one of these might look like. So you have bark sloughing off of a dead tree, and bats will actually get underneath this dead bark and form maternity colonies. And this is where Indiana bats will actually rear their young. Colonies of, or females in maternity colonies will rear 1 pup per year. And kind of as either a replacement or a supplement to these snags, artificial roosts are becoming more common as a management technique. And this is first of all to sometimes mitigate habitat destruction of these roosts, but also to expand availability in a certain area of roosts for bats. And also to facilitate research sometimes of those more cryptic species that are harder to capture. So more research is needed to understand the benefit and use of artificial roosts by bats. So we know that in looking at artificial roosts that roost temperature is important. Energy conservation is incredibly important, particularly for those female bats entering the reproductive cycle. And it's advantageous for females to avoid long bouts of torpor because this has been shown in several studies to impede embryo growth and pup development. So you'll see that 15 degrees Celsius was designated as our lower range for suitability for bats at our artificial roosts. Because again, research has shown high energetic costs for maintaining body temperature or going in and out of torpor below this temperature. Overheating is also an issue at artificial or natural roosts. So greater than 40 degrees Celsius was designated as our higher range or our high range or upper range for what we wanted to be seeing at these artificial roosts, because bats can undergo heat stress and overheating can cause fatalities at extreme temperatures. And also, if you're thinking about female bats in the maternity season, while it might be okay for male bats or non-reproductive bats to kind of switch roosts a bit more often, females are going to be caring for pups in the roost, or they're going to have a pup on the wing with them. And that's a little bit more difficult to be constantly switching roosts if these roosts are inadequate temperatures. So looking over here are our Brandenbark bark mimic roost structures. And so these were present at our study site prior to us beginning this study. And partially our study sites were selected based on the presence of these artificial roost trees. These structures do have a known history of maternal use. And what they're made up of here is a 7.6 meter utility pole. And then this is a polyurethane bark around the top. And so bats can get into kind of a crevice in between the pole and the top of the roost here. And that's where they're able to form these maternity colonies. So we're studying these roosts that were already available on our study sites. Our objectives for this section were to document temperature profiles and determine habitat suitability for these bark mimic roosts, and to determine where bats concentrated in greatest densities and if cluster placement or temperature difference impacted their use. I wanted to note that we are only focusing on Veterans WMA for this section of the study. And that is because of this layout of the 3 clusters of roosts at Veterans WMA, which bats are all using. And so we have up here our six roosts at our north cluster. That's under a kilometer away from our central cluster of 7 roosts. And then down here, there's our north cluster, which has 6 roosts, and that's less than a half of a kilometer between these 2 sections here. So moving on to our methods to look at temperatures of our artificial roosts, we put these hobo pendant temperature light loggers inside solar shields. That's what those plate looking things are here. And we set those on, or we built these data logger holster frames using conduit and a little bit of PVC pipe up here to hold both of our solar shields and our data loggers. And we were able to place these on top of these Brandenbark bark mimic roosts. So this is my coworker, Jim Bernard. He's actually taking off one of these holsters because this is at the end of season where bats have moved on and ceased using these roosts for the year. But you can see that we placed our data loggers at a northeast and southwest facing aspect to try to get both of those temperature scenarios in our recording. We put a total of 38 individual loggers out at Veterans WMA, 2 for each of the 17 roosts within the 3 or in the 3 clusters overall. And we're really operating under what is proposed here. We were not able to put temperature loggers under the bark due to disturbance concerns of the bats. We also placed data loggers on the northeast and southwest facing sides of a live shagbark hickory tree, which has been recorded to be an alternate roost. The live tree can be an alternate roost for bats in maternity colonies. And we put these at both the average height of our other data loggers within the cluster, and then we measured the diameter of the poles to get a tree that was adequate size in comparison to these artificial roost poles. And we did put 6 of these out, so 2 loggers, 1 tree at each of the 3 clusters. In order to assess the use of bats at these roosts, these guano catchers were installed underneath the artificial roosts here. And so Elizabeth Robinson actually created and put these out as part of her RAU project. So they were able to serve kind of a dual purpose in both of those studies. Guano was collected at these roosts every 2 to 3 days in different clusters, and when there was not collection at a certain cluster, the roosts were cleared off to maintain consistency. So in order to assess what factors were impacting out the temperature of these roosts, we modeled daily mean, minimum, and maximum roost temperature. So you can see those are our response variables for our 3 sets of modeling here. And then within each of these, we looked at the aspect, the cluster, or the aspect of the loggers, the cluster, and then seasonality, weather, or seasonality to kind of interpret how our roosts were changing throughout the season, as we would expect to do in a normal season. So we created generalized linear mixed models with a gamma distribution, and we created 5 model candidates in each of these sections of modeling and ranked these using AIC selection. And so for our model ranking here, we did have both of our cluster and seasonality model and our global model ranked with a delta AICC of less than 2. So they were both, again, competing models. And you'll note that we're only looking at the results for mean temperature because minimum and maximum came out almost identical. And just in the interest of time, we're going to only cover mean. And so we did conduct model averaging to pull out those informative parameters, and because of our competing models, you'll note that only our differences in seasonality had any significance for these models. So moving from late May through early August, the changes that we would expect to see in temperature, of course, these roosts are changing in temperature as well. You'll note that we did use 95% confidence intervals here, and that is because we kind of narrowed those down. We were having some issues with sign switching for our estimates in our different cluster variables, and we actually figured out that this was due to our estimates just being really weak for the differences in these clusters. That's kind of covered in that Gelman and Tuerlings and Gruber paper I have cited. So we were not able to consider the differences within our clusters as being significant. We also did not see any difference between our northeast and southwest facing loggers. So this is mean temperature on our y-axis, and then period across the maternity season on the x. We have our northeast facing and then our southwest facing here in gray. And you can kind of see those aspects look pretty much the same. The temperature ranges look the same throughout the season. We're not really seeing differences between the two there. Moving over to what our clusters looked like. Again, this is mean temperature here. We have our north, our central, and our south cluster. And we did kind of see that the north cluster was reaching a little bit warmer ranges throughout the season, and then kind of followed by our central and south. But again, we didn't see a significant difference in temperature here, and there was not a lot of kind of the average degree difference between the three clusters. And so cluster difference did not seem to impact temperature at VWMA or Veterans WMA. We also wanted to compare the artificial roost and then those live tree temperature loggers that we had put out on our shagbark hickories. In order to do that, we conducted the non-parametric Manu-Whitney test because of our differences in sample size there. And the only parameter that came out significant was our maximum daily temperature for these artificial roost and live tree temperature differences. And so you'll see that we have our roost in white and our tree in gray, our maximum temperature on the y-axis. And our roost did seem to reach higher temperatures in maximum temps throughout the season, which is what we would expect because our tree loggers are of course shaded. Those trees still have leaves on them, and our artificial roosts do not have leaves shading the roost and causing those cooler temperatures. Roost cooling did appear to be more of a threat to bats at Veterans WMA than roost heating. So we didn't have any roosts that exceeded our maximum temperature threshold of 40 degrees Celsius. Our highest log temperature was 33.4 degrees Celsius. Again, we don't know what temperature conditions look like on the inside of the roost, so this is our closest approximation. There have been other studies using bark mimic roosts that have seen higher temperatures or overheating temperatures in them. So but we can't assess that within our study. Our daily minimum temperature for individual loggers did fall below the minimum threshold of 15C on 648 instances in 29 days, and our lowest temperature was 5.5 degrees Celsius. So again, more of a cooling issue. Briefly, we did see that bats favored specific roosts. So these are indexes of our counts at each of the roosts. And so our south cluster by far had the most bats, followed by our central and then our north cluster. And what we're seeing here is that bats seem to favor, particularly in the north and central cluster, they seem to be favoring 2 roosts at each of these clusters. The south was a little bit more evenly distributed, but still kind of these 2 main roost poles that they're favoring for use. And anecdotally, when we were kind of looking at the conditions of these 2 roosts, there seemed to be one in each cluster that they used the most that was very, very solar exposed, very apart from vegetation. And then one would be kind of closer to the canopy edge and would kind of get a little bit more of that shade there. Bats also seem to spread across the WMA throughout the season. So we have our periods of collection on our x-axis. So these are different sections of Julian days where collections took place throughout the period, and then our mean guano count on the y-axis. And what you're seeing here is that at the start of the season, bats really favored that south roost cluster in black. And then as we move throughout later in the season, bats seem to spread more evenly throughout these 3 clusters. And they do seem to be more evenly spread at these higher temperatures that were logged at the end of season. This also may have something to do with the colonies getting larger, with juveniles being recruited into these colonies and possibly spreading out and finding new areas to roost in the WMA to kind of spread out from the main colonies. So with that, our conclusions for our temperature section are that cooler temperatures are a greater threat to bats at Veterans WMA. Reproductively viable adult females or juveniles were observed at all 3 of our clusters. So it seems that all 3 of these clusters at Veterans are suitable for reproduction. We also see that the varied roost placement at Veterans WMA, having some in those very solar exposed and some a little bit more shaded, seems to be successful in allowing bats to opportunistically choose roosts. For future studies, it would be important to look at the microclimate on the interior of bark mimic roosts and then do a comparison as well, if possible, between true snags versus artificial roosts. So for our final overall takeaways for each of our chapters, Chapter 1, maternity colonies of Indiana bats in Kentucky do demonstrate promising reproductive capability and condition. For Chapter 2, recapture records have demonstrated the ability of females to reproduce consecutively post-hibernation in white nose syndrome positive hibernacula, as well as to birth reproductively viable young. For Chapter 3, we know that maternity habitat is critical for pup survival, and this underscores the importance of variation in roost placement for opportunistic selection. With that, I would like to thank the Kentucky Department of Fish and Wildlife Resources for funding this project and the support of Zach Couch, Sunny Carr, Jim Bernard, and Tracy Hemberger throughout the 3 years of this project. I would like to thank my advisor, Dr. Luke Dodd, Dr. Kelly Watson, and our lab members, my committee members, Dr. David Brown and Dr. Cy Mott, as well as the management at Yellowbank WMA, Veterans WMA, and Bernheim Forest. And I would also like to thank a plethora of other agencies and organizations that provided support for this project as we were going about this process. And finally, to our bat capture volunteers, thank you all so much for spending your evenings driving across the state to meet us and make sure we had the help we needed for this project. So thank you all so much for your time. And with that, I will take questions.
Woo! Awesome. Good job, Michaela. All right. That was a lot of information. We knew that we would be coming up on the full hour, but we still have a few more minutes before 9 o'clock rolls around. Let's go ahead, and I think folks can kind of unmute themselves if you'd like to just go ahead and ask questions. We don't have to do the raise hand thing. I think with a group of this size, just go ahead and unmute yourself, and then feel free to just go ahead and ask away.
Michaela, I'll start. Very nice job. I thought it was encouraging that you're getting positive information about reproducing females. And I'm wondering if white nose syndrome has been around long enough that we're starting to get selection perhaps for individuals that are resistant to white nose syndrome. And maybe that's why reproduction is still working. Any thoughts on that?
Yeah, so that is definitely our hope here, is that we've kind of moved past that first wave where, you know, these mass deaths of bats from white nose syndrome has occurred. And we're hoping that at this point, bats are starting to, or the ones that are remaining are the ones that are able to kind of survive throughout the winter. There is some preliminary research out of the Northeast where, of course, it kind of came their first white nose syndrome, and bats do seem to be kind of exhibiting some level of resistance to this, where caves do not have as high of a fungal load that they did originally, and bats do seem to, there has been a little bit of genetic work to start to look into this, and very preliminary, they do seem to be starting to adapt. But again, that's kind of the hope here, and And as we move forward with white nose syndrome, hopefully we'll just continue to see some positive results.
Great, thanks.
Great, thanks.
Other questions? So Michaela, I have a question, and I mean, I know I'll have a chance to ask you more questions here in just a little bit, but something that maybe the audience, like maybe you can kind of provide a little bit more information. I don't know if any of the other agency folks might be able to speak to this, but how often are sort of like interstate banding records like that generated in such a fashion as like with what your chapter 2 is, like to show how bats are sort of moving across state lines? Is that something that KDFWR has previously been able to flag pretty easily?
It kind of depends on the year. So whenever those came out of that last big count, so there's those Indiana bat counts that happen every 2 years. So the really, really big colonies are counted every couple of years. And so after those counts, there are more frequent recoveries of bands from out of state. I think we've had Indiana recoveries before, but we actually, when we got those recoveries, we had some that we hadn't seen before. I guess they had just kind of forgotten to mention it or something like that. So it is kind of harder, obviously, just, you know, everyone's caught up in their own thing. So sometimes data doesn't get shared. So it's nice to kind of have that relationship where we were able to get some of those records. But that I know of, it's not incredibly common except for when there's those large counts.
Gotcha. And so just so the everybody that's listening in kind of knows too, can you just explain just briefly like kind of how surveys for Sedalis and Hibernacula lay out across that every other year kind of pattern?
Did you say how surveys progress?
Yeah, like the hibernation surveys, like you mentioned, that was a big year, right? So what's the kind of timing of that, like regularly, just for folks that may not be familiar with how that works within the bat world?
Right. So every 2 years, there'll be these larger counts where everyone that, or the states that are kind of part of the recovery of the Indiana bat will go into these caves in kind of very late winter. So we'll usually start in January and go into February. And basically it's a group of biologists that will go into the cave and try to get our best possible estimate of how many Indiana bats are in these caves. We actually do cave surveys every single year, but that I'm kind of talking about those bigger ones are every 2 years. And as I said before, these bats are clustered together sometimes by the thousands. And so they're, you know, you kind of have to use, like, there's different squares you'll take pictures, and you'll kind of take pictures, get things as quickly as possible. You'll actually go into a computer program and count individual noses of bats later to, which can just make your head spin because there's so many noses to count. But you're kind of going in, counting the small clusters, getting the big ones kind of as fast as an estimate as possible, taking down the notes, and then getting that full count after you get back when you've been able to count all of your individual bat noses later.
Awesome, thanks. Other questions from the
I have a question.
We still have a couple minutes.
Actually, is somebody else about to ask a question?
Well, David, I don't know, can you all hear me? This is Mike Armstrong.
Yep, you sound good, Mike.
All right. Yeah, just to speak a little bit, Michaela did a good job on the banding and the winter hibernation stuff. But the one thing I would add to that is that Kentucky is a little bit unique in that we do more surveys that occur every year than surveys from other states in the range of the Indiana bat. I think that's part of hibernation surveys, just because it's really not feasible to get them all done in 1 year.
Oh, you're breaking up, Mike.
But we do, like Michaela said, we do them with the large site. Any better at all now?
You're kind of fading in and out just a little bit, but I think the point you were making was that with Kentucky's hibernacular surveys, there's so many to count that is something that sort of spills across years. Is that right, Michaela? Is that kind of what that was being?
Yeah, so we will have some of the smaller priority caves in the kind of off years, and then the larger priority 1 caves will be kind of on that same 2-year rotation.
Hey, I have a question, Michaela,
Okay, Dave.
about banding. So, yeah, as a bird bander, I know that like bird bands are pretty tightly controlled by the federal government, and the data is as well. And you mentioned data sharing among bat banders, and I'm just kind of curious is how that data is organized and how bans are issued, and so would you even know for sure if somebody recaptured some of your bats elsewhere?
As far as like in the state goes, Kentucky Fish and Wildlife distributes the bans to folks that are doing surveys in the state, and then they have to go ahead and report that back. So we can get a pretty good idea of band recoveries within state that way. And then as far as the hibernacular accounts, we're of course very much involved in that in the state, so we can get those recoveries. It is a little bit harder in out of state situations because we're not directly getting that data. So it would just have to be, someone would have to probably reach out to us and let us know. But you mentioned it being regulated. You do have to, of course, have the proper permits and, you know, vaccination experience. So again, similar to bird banding, I'm sure it's quite similarly regulated.
Awesome. Any other questions from our audience? All right. Well, Michaela's going to have a whole lot more questions coming at her here in just a few minutes because we're about to wrap this up, and then we'll move into the thesis defense portion of the morning. Mike asked 2 questions here. So he asks, was the swabbing of bats done basically to look for the fungus PD? And then Michaela mentioned males at Bernheim. Did she see similar percentages of male at the other 2 sites? So maybe answer the swabbing question first, Michaela, and we'll tackle the Bernheim question.
So we didn't do any white nose syndrome swabbing. Our kind of impact of white nose syndrome for the 3 colonies was based purely on seeing those wing scores of greater than 0. So anything greater than 0, you're seeing that white nose syndrome impact. But we didn't do any individual swabbing and sending off tests. But some of those caves that bats were recovered at in-state have done some swabbing and sending off of tests to confirm that white nose syndrome positivity in the caves, like the B&O and then bat and saltpeter cave that I was talking about.
Gotcha. And then as far as at Bernheim, so we did have males, you know, incidences of that. So we talk about Bernheim versus the other 2 sites and incidents of male captures at the other 2 sites.
So for Bernheim, it seems like we definitely had higher captures of males really consistently throughout the 3 years. I think we had consistently higher captures of males. And I mean, at least as far as adults go, we did, when we had juveniles in July, it was a pretty even proportion of both female juveniles and male juveniles, which we would expect to see. But it's a little bit of a smaller colony at Bernheim Forest. And so it kind of seems like, whereas males in the other colonies, there just may have not, you know, they kind of have left the colony as they kind of should and are roosting singly. It seems that males at Bernheim, more than the other 2, we were catching them that were coming back to these colonies and just roosting along with them again. So males that are no longer juveniles, but they're coming back to roost in these colonies, I guess just because there's a little bit more room for them with that smaller colony at Bernheim.
Awesome. Okay, so let's we'll go ahead and wrap this up at this point. I guess I would just like to say one time, thanks to everybody for attending. And again, I threw shout outs to a couple of the feds that were on the call, but I know all of the various state folks, Sonny and Zach and Tracy and Laura and anybody else that I'm missing in the list. Thanks to all of y'all for calling in. I know that you have busy schedules in the COVID world also, so it's much appreciated. And then I guess on my end, I would just also like to say thanks one more time to Jim, Bernard, and to Tracy, and to Sonny, and of course to Zach for really facilitating this work. I mean, this really was a project, was like a fair split between KDFWR's efforts and the efforts coming from EKU. So I think it's a good example of how an agency and a university really are able to work together for a cool product. And thanks to Jim for all of his hard and long hours with Michaela in the field when Zach wasn't there or I wasn't there, or any number of other helpers. And then everybody else that showed up to track back so many nights, that's a big help too. So thanks to everybody. So I guess at this point, folks can drop off the call. I'm going to go ahead and stop recording here in just a moment, but Michaela and committee members, please stay on the line.